Okay, I'd just like to summarize some of the material from week 7, looking at oncogenes and their role in cancer. So basically, oncogenes are mutated versions of normal cellular genes, and there are some key mutations that can happen to these oncoproteins that give rise to a hyperactive protein. Okay, so if one of the two copies of the genes in your genome pick up this key mutation that makes it hyperactive, then it will dominate over the normal unmutated copy, and it will give rise to an altered growth phenotype. Okay, so there's plenty of mutations that occur in oncogenes that knock out the gene function, but there are a small subset of mutations that occur commonly in cancers, and these mutations occur commonly because a single point mutation can make the gene or the protein hyperactive over the normal copy that exists in the cell. So I'd like to but people first started to be able to study the effect of mutated DNA in cells once people were able to transfer DNA into cells and have it integrate and become functional within a cell. So once people were able to do these transformation experiments, they didn't rely on using viruses and identifying, human genes in tumor viruses. They were able to do experiments independently of the viruses and clearly establish that the source of the mutated DNA or the source of the phenotype was normal mutated DNA or mutated DNA from normal cells. So in these experiments, they chemically treated normal cells. They were able to generate mutations in normal genomic DNA, and then they were able to transfect some of this DNA into untreated normal cells. And this gave rise to small groups of cells which had increased growth phenotypes, and that when they transferred these cells into model organisms, they were able to induce these cells gave rise to tumors within the models. So effectively, these tumors are being caused by normal cellular genes that have been mutated. So clearly it really showed that cancer didn't come from outer space, didn't come from some, if you refer to that video from week 14 I showed about how, before the 1970s they thought cancer could have come from outer space. These kind of experiments clearly gave a very specific cause for cancers. They just needed to then identify what were the DNA sequences that had been mutated that were giving rise to these tumors. And then that starts to give you an understanding of what is causing the cancer and then how you can treat the cancer. So they've identified a whole range of these gene sequences that can be mutated, giving rise to altered growth characteristics. And these gene sequences are referred to as oncogenes. And there's been a whole range of these gene sequences identified. So to understand how these genes function, you need to have an understanding of the protein and an understanding of cell biology. So when they were looking at these gene sequences, and they identified the RAS gene as one of the sequences that transformed normal cells with an additional growth characteristic, they identified the RAS gene in these bits of DNA, and they identified a specific mutation, or a couple of specific mutations, that gave rise to a transforming principle of DNA. In other words, a DNA that was a bit more active, giving rise to a protein that's more active. So the point mutations in the DNA they identified gave rise to a switching what are they doing there? It's switching a glycine to a valine, or a valine to a glycine. I'm not quite sure. So the proto-oncogene is the normal cellular gene, so a glycine is being switched to a valine in that. So a single point mutation giving rise to an amino acid residue change, giving rise to a hyperactive protein. So once they'd realized that here's a key genetic change, we've identified it in a cancer, they looked at other cancers, and sure enough, these same mutations were occurring in the same genes in other cancers, from pancreas and liver and kidney and other samples. Also, they noticed that some genetic changes were chromosomal rearrangements, and what effectively these were doing, they were overexpressing another oncogene. So effectively, the coding sequence from the MYC protein was being switched to be downstream of an active promoter, giving rise to higher than normal levels of the oncogene, and therefore making it grow faster. So effectively, this wasn't a mutation in the coding sequence, it was switching the promoter of that gene to make that gene more active through translocation of the chromosomes. And so people identified the RAS sequence through these transformation experiments, and they also identified the SARC protein as being an oncogene as well. So the question is, how can a single point mutation have such a big effect on cells? And to understand that, you've got to have a bit of an overview of how cells regulate their growth. So simply, you have a range of growth factors external to the cell. These growth factors activate receptors on the surface of the membrane. They are transduced through the cytoplasm to turn on key sets of genes. Therefore, key growth factors can regulate growth or differentiation or migration, depending on which factor and which transcription factor is activated. Okay, so these normal cellular genes or proteins are key players in regulating cell biology, and these are candidates that can become oncogenic. So mutations that make these proteins overactive drive growth or differentiation much more strongly, if you like, than the normal state. So how does this occur? So we know SARC is an oncoprotein, and it's involved in regulation. It was when they started to look at how SARC functions, they realized it was a soluble tyrosine kinase, and they hadn't seen soluble tyrosine kinases before. And effectively, a tyrosine kinase has a catalytic activity that it can phosphorylate other proteins, therefore making other proteins change their function, and therefore activating and regulating things within the cell. And they did these studies using an antibody for the protein, and then the antibody would then get phosphorylated by the protein, and they realized, that it was phosphorylating tyrosine residues. They also, once they identified that SARC was able to affect many other proteins through the kinase activity, they realized that a lot of the receptors that were picking up the external signals also had tyrosine kinase activities, and that they could also become hyperactive and drive proliferation in the absence of growth factors. So this was a really key. Understanding Sark was a key to then understanding the receptors and understanding how the cells could grow in the absence of these growth factors. An example of one of these growth factors, which is potentially an oncoprotein, is the epidermal growth factor, EGF. And this oncogene can become activated by point mutations to become hyperactive. And this little diagram here shows that the EGF receptor has a region of homology with the soluble tyrosine kinase protein, and this is the tyrosine kinase domain of the receptor. So effectively, Sark and these receptors have the ability to phosphorylate other proteins and drive big changes within the cell. Clearly, there's quite a lot of receptor tyrosine kinases involved in growth and proliferation and differentiation, and mutations of these kinases occur often in cancers. So in some cancers, they've picked up mutations in various receptor tyrosine kinases, and that these mutations give the cells an ability to grow in the absence of external stimulating factors. Like I said, RAS was one of the first oncoproteins they discovered, and again, we try and rationalize how can a point mutation in the RAS molecule drive a cell to become cancerous. And I don't want to go into the detail of RAS activation here, but basically suffice to say that the RAS protein can exist in an active state and an inactive state, and it switches between these two states through interaction with a guanine nucleotide. So in the active form, the RAS is interacting with a GTP, and then it's through the hydrolysis of the GTP that RAS inactivates itself. So if RAS hasn't got the ability to hydrolyze that GTP, then the RAS will stay active. So a point mutation that affects the hydrolysis of GTP means that RAS stays active for longer, and therefore it drives growth for longer. There was some debate in the literature for a while whether RAS, which is an oncoprotein, and EGF, which is an oncoprotein, that there was some debate about whether RAS was acting upstream of the receptor. In other words, RAS was leading to the release of factors which were activating the EGF, or whether RAS was acting downstream of the receptor. The textbooks gives you some compelling evidence that RAS acts downstream of the receptor, and we've discussed the fact that RAS is downstream of the receptor. So clearly RAS is an oncoprotein, meaning that there are two copies of the RAS gene in the genome. If one copy of the RAS gene picks up a key mutation, it will make the RAS protein more active. So even though there's the normal RAS protein in the cell, the hyperactive version of the RAS will dominate and drive the phenotype of more growth. The normal acting RAS doesn't compensate for the active RAS. The active RAS dominates and drives proliferation. So you don't need to have the same point mutation in both copies of the gene. A single gene can pick up a single point mutation, leading to an altered growth state. So I guess if you look at the RAS protein in a bit more detail, it shows here that those point mutations that were occurring in H-ras in tumor cells were point mutations that affect the hydrolysis of the GTP. And yes, so that would make it more active. So, so in this lecture, we looked at oncogenes, the characteristic of mutation in the oncogenes, and where some of these oncogenes fit in terms of general cell biology. Okay, hope that was useful. Thanks.